Hello everyone, I am Dibyendu Samanta from IIT Kharagpur, and I am discussing now the fourth lecture of module 2. Today I am going to discuss about time-space correlation and fidelity of transcription and translation. During the last lecture, mostly I was discussing about the basic mechanism of protein synthesis. So during that time, I mentioned that there are much more complex things present there. So today's in this class, I will discuss some of those complexity, some of those fundamental understanding, but still they are much more complex. So particularly, I will concentrate on logistics of transcription and translation in both prokaryotes as well as in eukaryotes, and also fidelity of transcription and translation. So those are very important steps in central dogma. Now, first, if I start with logistics of transcription as well as translation in prokaryotic systems. So here I am going to discuss about prokaryotic system, such as in case of bacteria. Most of the studies actually conducted in E. coli, because it's a beautiful model organism, non-pathogenic E. coli, like they are the model organism in lab. So that's why. So now, as the name suggests, that prokaryotic, that means they don't have nucleus. Let's see where those steps are happening. So if I draw, this is a bacteria, this is E. coli, Escherichia coli, E. coli, a bacteria. So bacteria, they have, particularly E. coli, many other bacteria, like they have just one DNA molecule, and that DNA is also circular in nature. So as a result of that, they have circular DNA. So if I say this is their DNA, now DNA is double stranded, so better put this double stranded something, schematic diagram of double stranded DNA. So now I am discussing about transcription and translation. So that's why I kept this portion little bit in a different way, so that I am going to show here that the transcription is happening. So then during transcription class, I told that transcription will be happening like this. So this is 5' end of mRNA, and this is 3' direction. It's going 5' to 3' direction. And here you can imagine that you have here RNA polymerase, the enzyme responsible for transcription. Now, since bacteria, they don't have nucleus, so they have just one compartment, the cytoplasm. So DNA is present there, and the transcription is happening. And during the last lecture, I mentioned that mRNA is the template for translation. So if I consider that this RNA, which is being transcribed now, if this is an mRNA, so it can immediately start, the translation can be started now, because the template is already available here, and the ribosomes are present in the cytoplasm. So you can see that ribosomes are present here in this bacteria. So these are ribosomes. So as a result of that, ribosomes can bind here to this already available mRNA, which is present in the cytosol. But this mRNA is still being synthesized. Still the transcription is going on. So as a result of that, what I am trying to say in case of bacteria, in case of prokaryote, the transcription and translation, they are coupled process. They are happening together. So transcription, translation, they are coupled together because all the necessary ingredients are available in the cytosol of bacteria, so that even before the completion of the transcription, the mRNA can still be translated by the ribosome. So as a result of that, ribosomes, they are going like this. So now I mentioned particularly here that mRNA synthesis or the transcription is processive. That means one RNA polymerase is synthesizing the whole gene, the whole mRNA is being synthesizing. Now that mRNA can be translated by multiple ribosomes. So that is kind of, we are amplifying the signal also. Amplifying the signal means from one mRNA we can have multiple proteins. So as a result of that, if I just draw here, this is one mRNA. This is just one mRNA, 5 prime to 3 prime. So ribosome started translating this mRNA here, and after some time this ribosome will be here and this protein is being synthesized here. So this is our ribosome, which started translation little bit before than this ribosome. So here protein is very small, here protein is very big now, because it is already synthesizing protein kind of little bit longer than this. So this is also another ribosome. But what I am trying to say, this mRNA which I draw here, this is mRNA, but this mRNA is still being synthesized in E. coli. So why I am telling this? That means there should be some kind of relation between the speed of transcription and translation. So for example, if the translation is so fast here, if translation, the process of translation, that means how ribosome they are reading the genetic code or the codon present in mRNA, if that is much faster, then this ribosome will hit here in the transcription complex. So it will create problem. So that's why I am saying that there is some kind of relation in their speed also, so that everything is being, in optimally in our living system. So as a result of that, what I mean to say here, that in case of bacteria, in favorable condition, in optimal condition, for example in E. coli, the rate of transcription is, you can consider, I would say 51. This is not so specific, I am just taking this number so that it will be easy to explain. So 51 nucleotide per second, okay, 51 nucleotide per second. It is kind of varying from, I would say, from 40 to 70, 80 nucleotide per second in E. coli, depending on the situation. So if this is the rate of transcription, then what should be the rate of protein synthesis? Scientists observe that the rate of protein synthesis in bacterial system is approximately 15 to 20 amino acids per second. So for example, here I would say I can just put, for example, 17 amino acid per second. This is not so specific, but for explanation I am saying 17 amino acid per second. Then why I put 17 amino acids per second? Because I already mentioned in the last lecture that 3 consecutive nucleotides, that is called codon, that encode 1 amino acid. So as a result of that, if this is the pace, this is the speed, then this ribosome will never hit this transcription complex. So everything will be happening smoothly, right? So that's why here I am saying that 51 nucleotide per second, the rate of transcription, and rate of translation in that case 17 amino acid per second. So this way transcription and translation, they are coupled process in bacterial system. So now we should look into eukaryotic system, in our body, in human cell, what is happening. So this is now I will explain in eukaryotic system. So it can be plant, it can be animal, or it can be human cell. That doesn't matter. Eukaryotic cell. So eukaryote means they have true nucleus. They have nucleus. Let's see what those steps are happening. So DNA is present where in eukaryotic cell? It is present inside nucleus, not in the cytosol. So as a result of that, now if I draw a very big eukaryotic cell, so it will help me to explain the stuff. So this is some eukaryotic cell, right? So eukaryotic cell, this one, and it has nucleus also. Nucleus, this is a different compartment. This is, getting separated from the cytoplasm. This is cytosol. So nucleus is present inside the cell, but it's a different compartment. And nucleus harbor those DNA or deoxyribonucleic acid. So DNA is present here in the nucleus. It's linear DNA in eukaryotic system, and in case of human in different species, they have different number of DNA molecules present in each cell. Now, after transcription, right? After transcription, we will get RNA. It can be mRNA, ribosomal RNA, transfer RNA, everything. So I'm just mentioning here mRNA. I mentioned during transcription class that that this mRNA, they need to be processed. That means addition of the 7 mg cap at 5 prime end. So here 7 mg cap, and here at the 3 prime end poly-A tail, and also splicing, the removal of intron. All those steps are happening inside the nucleus. Processing is happening. Processing of pre-mRNA. This is called pre-mRNA. So processing of mRNA is happening, and finally we have mature mRNA with 7 mg cap, poly-A tail, and introns are already removed. So those mRNA, they will come out from the nucleus. So nucleus, although I told it's a different compartment, but if you see, they have some, they have some like pore there called nuclear pore. So they have some pore here. So that things can go inside and outside. So as a result of that, this mRNA will come out through this pore outside. Outside means this cytoplasm. This is now mRNA coming into the cytoplasm. mRNA export from nucleus to cytoplasm. Why it needs to come? Because ribosome is actually waiting at cytoplasm. Ribosomes are present in cytosol. So here ribosomes are present. So these are our ribosomes. So now these ribosomes, whatever I told that 30S, 50S in case of bacteria, and 40S and 60S, so those are different size particle based on their centrifugal force, like they can be separated differently, right? Those are called Svedberg unit based on scientist name. So those subunits, they are available in this cytoplasm freely. They are not together. Only during the translation process they get united, like 30S and 50S, they get united and they carry out translation in bacteria. Similarly, 40S and 60S, they get united to make the ribosome and that translate protein inside eukaryotic cell. Now, this ribosome will start translation from 5 prime to 3 prime direction. I'm not showing here the cap and the poly A tail, but that should be present here. So this way translation should be happening in cytosol. Here I would like to mention one thing, that translation is happening inside, or I would say in the cytoplasm, right? So that's why after translation you will get protein molecule in cytoplasm. Now the replication, transcription, everything is happening inside the nucleus, and that requires enzyme, DNA polymerase, RNA polymerase. So that means we have very complex system, so that the required enzymes, which is actually required inside the nucleus, after translation they will be transported to the nucleus again. For example, RNA polymerase is being synthesized at the cytoplasm, then it will be transported into nucleus so that it can transcribe RNA molecule, right? So that's why I told that it's very complex scenario. Similarly, another level of complexity is like even the ribosomal RNA, which constitute ribosome along with many different proteins. Ribosomal RNA will be synthesized also inside the nucleus because it's the result of transcription. So because of that, it is very interesting to know that ribosomes are assembled, ribosomes are generated inside the nucleus itself. So ribosome particle, for example, in this case, 40S and 60S particle of ribosome in eukaryotic system, they get assembled inside the nucleus, and then those ribosome particles, they are being exported in the cytoplasm, and they do translation here in the cytoplasm itself. So if you see all those things, those are actually really complex, and everything is happening in a coordinated fashion, so that like life is moving very, very smoothly. But all those things are because of many like years of evolution, and it makes a very robust system. So now I would like to show you something else also here, that time, space and correlation between transcription and translation. Little bit I already mentioned, like when I was talking about in reference to presence and absence of nucleus in prokaryote and eukaryote. So here this is one important and real diagram I have taken from this paper. So this is very very interesting to understand this complexity, whatever I just mentioned, but this is the experiment they are showing. So in this figure, if we concentrate, you will be seeing a very very light thread-like structure in this figure. I am just drawing over it so that it will be easy for everyone of us. So for example, if here, see, here you have a very thin line here. It's going like this, going like this. So this thin line is nothing but it's, what is this? This is DNA. This is the DNA. I will explain everything is mentioned here, right? This is DNA. Now, in this DNA, you can see some small, this is also not very prominent here, 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 here. So those are RNA polymerase. This figure is electron micrograph, very high resolution image of during translation event in bacterial system, in prokaryotic system, right? So this is RNA polymerase. So RNA polymerase is actually transcribing RNA. It's transcribing RNA, it's making the RNA. DNA is the template in this case. Now, then what are those particles? So these particles. So these are nothing but ribosome. They are ribosome. Ribosome. And this ribosome together, like many ribosomes, they are together, they are attached to a single mRNA. As I mentioned during the explanation of coupled transcription translation in bacterial system. So these so many ribosomes attached to the same mRNA is called polyribosome. So multiple ribosomes they are together, they are translating the same mRNA. This is polyribosome. So now the question is, you can see it's the same DNA, single DNA molecule, right? And we have we are showing here multiple RNA polymerase. So this RNA polymerase, for example, here, after some time, it will transcribe, and it will go in this direction, and it will reach here. Then the length of mRNA will be more, and more ribosome will be attached here. So this is like, this right side here. From this position, from this side, transcription started. Why? Because you can see the length of mRNA is small here. That's why less number of ribosome here. When this is going in this direction, then the length of mRNA will increase. As you can see here, the length of mRNA is more here, and more ribosome attached to this, this mRNA. So as a result of that, from this figure, we can say that transcription is happening in this direction. Then what is the direction of translation? So translation direction should be here ribosome, they are going like this. Ribosomes, they are reading the mRNA in this direction. So because of this experiment, because of this data only, we were able to understand this coupled transcription translation, which I already explained. But this is the actual, experiment from where we came to know all of these things. Now, the same thing, this is a reconstituted image for all of us. So we can, if our eye is not very specialized to see this kind of, this kind of electron micrograph, this is much easier here. So as you can see, this is our DNA, and this is RNA polymerase, and direction of transcription, whatever I already explained. And then you can see that like here the length of this mRNA is not that big yet, but here the mRNA is much bigger, and so many ribosomes are attached together, and coupled transcription and translation happening in this way. But now if I discuss kind of similar thing in eukaryote, this coupled transcription translation cannot happen, because as you know that transcription is happening inside the nucleus and translation is happening outside the nucleus. So this is not a coupled process. So as a result of that, if I draw, this is an mRNA. So this is our 7mg cap, and here we have poly A tail, and this is mRNA of eukaryotic system. So when it is in cytoplasm, then it will be translated. But to increase the efficiency, we have a different kind of strategy. So in case of bacteria, like transcription and translation, they are coupled process. Even before the synthesis of the full mRNA, translation started. So translation rate and transcription rate, they are kind of matching in such a way so that it is a very efficient process. But in this case, if you see this mRNA, in many times they can make some kind of loop-like structure like this. So here you have 7mg cap at the 5' end, this is our 5' end, and here we have poly A tail at the 3' end. How they form this loop-like structure? Because some proteins are there. So specific proteins are there. Somehow they interact with each other here. Some protein molecules here, like poly A binding protein, like this is poly A tail, so poly A binding protein, and some eukaryotic initiation factor. So they will bind together here, and that way they will make this kind of circle. But what is the advantage of this? Advantage is, so when ribosome they are translating this mRNA, they are going in this direction, right? And what will happen when they are done with translation process? when they are here, then they will be separated. I already told ribosomes during translation, they united in this manner, but otherwise those two subunits remain separated. So here, when the translation is done, that means when they encounter the stop codon, then the translation is done. So as a result of that, during that time, those ribosomes will be splitted, free ribosome particle. But now the 5' end is very close by, so again they will start. So that way the efficiency, the effectiveness of translation is much more. So the ribosome concentration, the particle of this 40S and 60S subunit will be more in this region, because they are getting dissociated at the 3' end, and again they will quickly bind to the 7mg cap, and that way it will start the translation. So that's why it is very, very effective process. So here you can see the whatever I explained is same thing here. This is poly A tail, and this is poly A binding protein, which is binding to eukaryotic initiation factors, like 4G and 4E. Those are the kind of name. You don't need to remember those things, but that way it makes some kind of closed loop, and that will increase the efficiency of translation. But now, while I was discussing that, I told that when it will encounter the stop codon, then the translation will stop. During the translation lecture, I forgot to mention that. But how? How it will stop? Because, like, very simply, we can imagine that whatever those 3 stop codons I mentioned, like UAA, UAG, and UGA, maybe matching tRNA is not there. The matching tRNA means the anticodon loop with the matching sequence is not present in our system. So as a result of that, no tRNA is available. They cannot bring any specific amino acid. And during that time, one specialized protein called release factor, it came into place, and it terminated the protein synthesis. That's all. Now, here some more interesting complexities there. I'm not going into all the details of that, but just to highlight here. During translation, another interesting question is which one is moving, whether the mRNA is moving or the ribosome is actually reading every 3 nucleotide, that means codons one after one. Which is true? Maybe both are right, because as you know that ribosomes are present in eukaryotic system, they can be present freely in the cytosol, in the cytoplasm, in eukaryotic system. Similarly, inside eukaryotic cell, there are so many organelles. One of those name is ER. ER stands for endoplasmic reticulum, right? Endoplasmic reticulum. This is kind of sac-like structure present in eukaryotic system. So on the surface of endoplasmic reticulum, many ribosomes are attached together, and especially they synthesize proteins which will be exploded from the cell, which will come out from the cell. There are many proteins, they should be secreted outside the cell. So they are being synthesized by ribosome, which are bound to the endoplasmic reticulum. Then which one is moving, mRNA or the ribosome? So those are kind of complex and challenging question. But anyway, so now this is the again the last slide of this presentation. So here I am trying to explain that time-space correlation between transcription and translation from an evolutionary perspective, not just inside one cell, like whether it's eukaryote or prokaryote, whatever I mentioned, from an evolutionary perspective, I want to say. Since I already mentioned that translation is so fundamental process to make all protein in our body, but it is being catalyzed by RNA. And at the same time, RNA can act as genetic material also. So scientists presumed that initially when life formed, at the very, very beginning, maybe we had RNA-based system where RNA can go into RNA. So it can act as our genetic material as well as it can catalyze the reaction. And over the time, evolution of RNAs that can direct protein synthesis, right? So as you can see, finally RNA, they are making protein. So this is some like, it will not, happening in just a year or two. This is evolution years after year, thousands of years. Like that way, it can be like this kind of a complex system. And present day cells, what we see today, that is the evolution of new enzymes that synthesize DNA and make RNA copies from it. That replication, transcription, translation, everything is happening properly. But now if you see, as I already mentioned, this step, the main catalysis is done by RNA, not by any protein enzyme. So this is very interesting aspect. And I want to mention, like, here additionally, that whatever I mentioned, that bacteria, nucleus, like bacteria, they don't have nucleus, and eukaryotic cell, they have nucleus, and whatever happening, transcription and translation. Inside, like eukaryotic cell, some organelles are present, right? For example, mitochondria is present in eukaryotic cell. Inside mitochondria, they have their like small, DNA, small genome. This is very surprising, right? Not other organelle they have. Like chloroplast in plant and mitochondria in plant and animal, they have their own DNA molecule. And that get also transcribed to make some important RNA, and that makes some protein also. And this is the reminiscent, this is the idea that maybe at the very beginning of this, during the evolution, some eukaryotic cell or some bigger cell, they actually ate those bacteria, and they are still maintaining inside the eukaryotic cell, and they have their protein synthesizing machinery, they have the transcription machinery, but they are also relying on the cytoplasmic protein also that can go from cytoplasm to mitochondria to chloroplast, all those things, but still they are maintaining this kind of situation that inside mitochondria, inside chloroplast, transcription, translation is happening, and they are making very important product. All those things are very complex, but if you are interested, you can refer some, many textbook, and you will get all this kind of, idea. So this is called endosymbiont theory, whatever I just mentioned, that bacteria, they went inside the eukaryotic cell, and they are still present, and they are doing very important work for us in the present day cell. Now, I am going to discuss about fidelity of transcription and translation. So, if we see all these three steps, that means replication, transcription and translation, you will see during replication, we have rigorous proofreading machinery. That means, I would say DNA to DNA, that is the steps we called replication. So, during replication, the proofreading activity is very much stringent, because DNA is kind of archival copy. So, whatever information present there, we have to maintain that information in a proper way. Like any mistake will only lead to a wrong mRNA followed by a wrong protein. So, as a result of that, although during replication some minor mistakes may happen, but we have very good machinery to correct that mistake. So, there are a lot of different types of machineries available inside the cell, so which will rectify the mistake. But in the next step, when we are getting RNA, so particularly I am mentioning messenger RNA through the process of transcription. So, during this stage, although some level of proofreading activity is there, some kind of correction activity is there, but I would say it is not like comparable to replication. Here some minor mistakes may happen, But that is not a big problem, because anyway, from one gene we are getting multiple mRNA molecule. So now if one of them contains some kind of mistake, then we have very good machinery so that mRNA can be destroyed soon also. On the other hand, if we say like from mRNA we are getting finally proteins, right? So this is translation. If because of some mutation, some wrong nucleotide present in mRNA, that means something, some problem in some codon, so that will code some a different amino acid in protein, and because of that amino acid, if the protein structure is not proper, if it is not attaining its native conformation, the protein will also be destroyed. So as a result of that, what I'm trying to say, that DNA is our archival copy, so where we have to keep our information safe, and then mRNA is just like a sticky note. For example, in every lecture, I am kind of recommending one textbook, for example, Lehringer's Principle of Biochemistry. Now, if something is wrong in the textbook itself, then all students who are actually following this textbook, they will learn something wrong. So as a result of that, the textbook Lehringer's Principle of Biochemistry should be very much correct form, right? So similarly, the information in DNA, it should be very much in its correct form. And now when you are actually writing something from this textbook in a sticky note or on a piece of paper, when you are writing, maybe by mistake you wrote something wrong, nothing will happen. Only you will just destroy that piece of paper and again you will write, right? So that way, I would like to say the transcription and translation, although we have some kind of machinery to take care of it, but that is not that much essential that you have to make it exactly, just like DNA, how it is getting probated. So that's all about this fidelity of transcription and translation. Briefly, I just discussed, and that's all. These are the textbook you can follow, and thank you very much.